Hi my dear students, welcome back to my class. SSLC 2022 Science Examination Preparation Final Revision Video Series 6. I welcome you all to the video series 6. So in this particular video, I am discussing about the most expected and important questions from the lesson Heredity and Evolution. Dear students, most of you will find this lesson as a difficult one. Actually, this lesson is very easy because only few concepts are repeatedly asked in every examination from this particular lesson. So as I go ahead in this video, you will understand the important concept that you must study and you must understand from this particular lesson. And if you are visiting my channel for the first time, don't forget to subscribe my channel and also share the videos maximum with your friends. And if you have not yet watched the previous Final Examination Revision video series, the link of those videos and also the physics video series link will be given in the description box below. Kindly find those video links. So when we look at heredity and evolution, so for this particular lesson, minimum 5 to 6 marks is expected. From this particular lesson, minimum 5 to 6 marks is expected in your final examination. When we look at both model paper and also the preparatory paper, we can see 5 to 6 marks is been allotted for this particular lesson. So let us understand the important questions. So the first question is, explain the two methods to estimate the age of fossils. Very important, dear students. When we look at the age of fossils, see, we can estimate the age of fossils in many ways. So as they have asked the two method here, the first method I would like to suggest is the relative method. So what is meant by relative method? See, if we dig the earth, we will get the fossils. What is meant by fossils? Fossils are the dead remains of the living organisms from the remote past. That means whatever the living organisms which were present on the earth several years ago, they got buried under the earth. Okay. So when we dig the earth, we get those fossils. That means the traces of those organisms. So the fossils we find closer to the earth's surface are the more recent one than the fossils we find in the deeper layers. So this is the one method which we can adopt to find adopt to estimate the age of fossils. So which we call it as relative method. Second method is by detecting the ratios of different isotopes of the same element in the fossil material. So we will detect the ratio of the isotopes. That means those organisms contains the carbon. So the amount of carbon present in those organisms can be estimated. So these are the two methods that we use to estimate the age of fossils. So you have to note down, it's a very important question. Second question, no two individuals are absolutely alike in a population. Why? First of all, you need to understand this question. So they are saying no two organisms are looking exactly identical. Alike is nothing but exactly identical. Why? What is the reason? See, it is because there are variations in DNA due to the crossing over and also the recombination during the DNA copying process. And when does the DNA copying process takes place? When there is fertilization during the reproduction process, as a fertilization takes place, that means the fusion of male and female gamete takes place. During that time, the chromosome from the father and the chromosome from mother, they will combine. So as they combine, what happened? The DNAs also will combine. So as a result of the recombination of DNA, what happened? We can find a variation in the newly born organism. So that is the reason no two individuals are looking absolutely alike in a population. Hope you understood this question. So now let us go for the next question. Mendel crossed the plants bearing the red flower, that is capital R, with the plants bearing the white flowers, that is small r, and produced the progeny from them. You have to know the basic words here, dear students. What is meant by progeny? See, progeny is nothing but the next generation offspring or the next generation baby we will call. See, how do you identify whether, first of all, it is a monohybrid or dihybrid? That is the first thing you have to understand. See, in my crash course videos, I have already explained every individual topic based on the heredity and evolution lesson. See, if there is only one particular type of trait What is meant by trait? Trait is nothing but the character. You can see here, Mendel crossed the plants bearing the red flower. What is the red here? Red is nothing but it is the characteristic of a color. So here you have the contrasting character. So the red is the one which is having the contrasting character as white. Okay, if there is only one particular trait, then we can consider that as the monohybrid cross. So first and foremost, you have to get that idea. So here, the plants with the red flowers obtained in F1 generation were different from the plants with the red flowers of the parental generation. Why? Explain the reason. So that is the question they have asked. So that is what the question they have asked here. Okay. So here, see the parental generation, the plant bearing the red flowers has both the dominant traits only. You can see, if you observe the red flower, there is capital R, capital R. Right, and the plant bearing the white flower have both, that is recessive traits only, that is small r and small r. But when we cross pollinate the parents which is having capital R with the small r and small r, what happened, in the F1 generation, okay, a copy will be inherited, a single copy of allele will be inherited from both the parents, that is one capital R and another small r. So you can observe here, so here it is represented. You can observe this schematic representation. So red flower parents has capital R, and whereas the white flower parents has the small r. When they cross pollinate, you can see, so from the first parent, one letter is been removed. We will call it as allele. And from the white color flower, that is from this particular parent, we are taking small r. So as a recombination, we got capital R, small r. Correct. So now this is not completely identical to the parents who are present in the F1 generation. Correct. So that is what they are exactly asking. The plants with the red flower obtained in the F1 generation were different from the plants that we got in the F1 generation. Why? Because when we look at particularly in the F1 generation, it is the combination of both the parents. But the flower is red here because the dominant trait dominate over the recessive trait. If you have this much concept clarity about the monohybrid, okay, you can easily get the full marks if they ask any particular trait in the examination. Okay, so you have to note this. It's a very important thing, dear students. So moving to fourth question, see again I am asking the one more question based on the CHAKKAR board only, because sometimes the CHAKKAR boards are given for 3 marks, sometimes it is given for 4 marks. You can expect either monohybrid or the dihybrid. Okay, so the next question, the plants bearing the round yellow colored seeds are self-pollinated with the same plant represent the result obtained in the F2 generation of dihybrid cross. With the help of the CHAKKAR board, mention the variety of plants obtained in the F2 generation. See, they are not bothering about the F1 generation here. They are particularly asking about the F2 generation only. But in the class, you have learnt both about F1 and F2. Some students find difficult. Without knowing F1, they cannot write F2. Okay, there is no problem that in examination, you can also write F1 along with the F2, dear students. Even though they ask F2, if you are finding difficult to write directly F2, please solve with F1 only. Okay. So, as in the question they have given the resultant of the F1 generation, that is round yellow with the alleles capital R, small r, capital Y, small y. So, this is the offspring that actually we got in the F1 generation. So, on segregation we get capital R, capital Y, capital R, small y. So at this point of time, I would like to inform you, those who do not know to write this particular CHAKKAR board, I have taught in a separate video called how to write the CHAKKAR board easily. You can find the link in description box below. Do watch that video. Okay, here I am only explaining the questions. Okay, you can find that video and understand how to write the CHAKKAR board easily. So when on segregation, we get capital R, capital Y, capital R, small r, capital Y, small r and small y. So when we write it in the table and when we self-pollinate between these parents, finally we will get 9 round yellow, 3 round green, 3 wrinkled yellow and 1 wrinkled green. Very important, dear students. This is the phenotypic ratio of F2 generation. See, in the examination, to write the checkerboard and to write the phenotypic ratio and to write the offsprings which is obtained in the F2 generation, the marks is allotted. So do not miss to write anything. Okay, this question is very important with respect to dihybrid checkerboard. To next question, what is evolution? Explain the 3 evidences for evolution. See children, first of all you have to know or you have to have a basic idea about evolution. See, evolution is a process which does not happen within 1 or 2 days or 1 or 2 months or 1 or 2 years. It takes longer period of time. Take an example, see, we are the human beings, we have evolved from the chimpanzees, and for the evolution, to get this particular body which we have now, so this evolution has taken place for a longer period of time. It does not happen within 1 or 2 years. Okay, so phenomenon of gradual change of an organism from the simple form into a complex form in a long period, we will call it as evolution. I think you got a basic idea. So there is a next question in the same question, that is explain the 3 evidences for the evolution. So they're asking the evidences. How can you give the evidences that evolution has taken place? First one is by looking at the homologous organs. See, what is meant by homologous organs? The organs which have the same basic structure but modified to perform the different functions, they are called as homologous organs. Okay, and by looking at these homologous organs, they will provide the information that the organisms of different species might be evolved from the common ancestor. So for this, you all can look at the internal body structure of a chimpanzee, human being, or we can take some tiger. So the internal structure of we all is somewhat similar, but the functions we perform is different. Hence, we have some connection between each other. Understand? So, by looking at the homologous organs, we can say that evolution has taken place. And even by looking at the analogous organs, what is meant by analogous organs? See, analogous organs are the organs which have the different basic structure, but they perform the same function or the similar function. So, for this, you might have understood about the wings of birds and also the butterfly. When we look at those organisms, their body structure is not similar, but flying is the same function which they both have. Okay, so by looking at the analogous organs, we can say that it is also that we can take some of the evidences for evolution. Okay, and by looking at the fossil records, see the dead remains of past organism which we find underneath the rocks or deep under the layers of the earth, we will call it as fossils. So by studying the different organisms, fossils, their traces, they help us to study the missing link between the different organisms. Okay, they help us to understand the sequence of evolution. Okay, so this is another way that we can find, there is evidences for evolution. Okay, so these are the 3 different evidences that we can consider that evolution has taken place over a longer period of time. Fine. Moving to next question. Name the 4 factors responsible for speciation. Dear students, they can also ask about what is meant by speciation. How can you define? See, speciation means origin of new species from the existing species. That means already some kind of organisms are there. From those organisms only, new organisms are being born. Okay, so origin of new species from the existing species, we will call it as speciation. And which are the 4 factors that are responsible for speciation? See, the factors responsible for speciation are: one is the natural selection, second one is the geographical isolation, third one is the genetic drift, and fourth one is the reproductive isolation. The 4 are the factors which are responsible for the speciation. Okay, so moving to 7th question, how does relative method help to determine the age of the fossils? See, again and again, related to fossils, many times the questions are arised. Okay, how can you determine? The relative method is helping you to determine the age of fossil. Earlier, I just told about how to estimate the age of fossil, but now I am pointing about only by looking at relative method, how can I estimate the age of fossil? I already told you. So fossils which are found on the upper layer of the earth are the most recent fossils, and the fossils which are found underneath the earth, under the deep layers of the earth, they are the most older one. Okay, so this is what we will understand through the relative method. So with the help of relative method, we can also estimate or determine the age of the fossils. So eighth question, why the experiences of an individual during its lifetime cannot be passed on its progeny and cannot direct the evolution. So before understanding the question, few concepts you must know here, dear students. What is meant by inherited trait? What is meant by acquired trait? See, the trait which is passed from your parents to you is inherited. For example, if your father and mother is having the black color eyeball, and if you have the black color eyeball, or if your father and mother is having the free earlobe, and if you are having the free earlobe, that is the inherited trait. If your father is so genius, but you're not so genius as your father, so then I can consider it as an acquired trait because it cannot pass it from one generation to another generation. So main concepts you have to know. And these acquired traits cannot pass from your father to you or you to your next generation. That is being obtained during their lifetime. Understand? So the experiences of an individual during their lifetime are called as acquired traits, and these are the changes which are present in the non-reproductive tissues of the individual, and they do not affect the DNA of the germ cell of that individual. So, as in the reproduction, only the germ cells participate, the changes in the non-reproductive tissues are not passed from one generation to another generation. Okay, therefore, it cannot direct the evolution. Understand? Yes. Moving to next question. The ninth question is, chromosomes inherited from the father determines the sex of the child. Or this question can also be asked in another way. Sex of the child is determined by the father. How? So the same question. Okay. But they asked the same concept based question. They have asked in a different way. Okay. So but answer remains same. See, the sex of the children is decided by the chromosomes present in the father's body, because father has the mismatched pair of chromosome. He has both X chromosome as well as the Y chromosome. Okay, so male has mismatched pair of chromosome, but the female has the perfect pair of chromosomes, that is the X and X chromosome. Okay, during reproduction, if a sperm carries X chromosome, okay, and when it combines with the chromosomes present in the mother, that is in the female body, the resultant offspring will be the girl child. If father releases X, in simple words, the resulting baby will be girl. Similarly, if the father releases Y chromosome during the reproduction, so then the resultant baby will be the boy baby. Very simple, dear students. You can write it in your own words. So here is also a schematic representation. Those who cannot, describe it in words, you can also write this schematic representation. You will get the marks. Okay? Yes. So this is an expected question. So moving to 10th question. What are fossils? Already I told you about fossils. Fossils are the preserved remains of living organisms from the remote past. Why I say remote past? Because it is, the product of several years, several million years or several thousands years ago. Fine. So moving to 11th question. What is the difference between homologous organs and analogous organs? Already I have told you about homo and ana. Homo is nothing but first of all same. So when I talk about homologous organs, the organs which have the same basic structures but modified to perform the different functions, we will call it as homologous organs. Similarly, when I talk about analogous organs, the organs which have the different basic structures but they perform the similar functions, we will call it as analogous organs. Okay, so it's a very important, you must know the difference between the homologous organs and also the analogous organs. Next question, write the differences between the sex chromosome of a man and also the sex chromosome of a woman. Okay, here they're asking about the chromosome, not the gametes, because most of you will write about the sperm and the egg. They're not asking about the gametes, they're particularly asking about the chromosomes. So when we talk about the sex chromosome of a man, so sex chromosomes are denoted as X and Y in case of man. But when we look at the woman, so sex chromosomes are denoted as X and X in the case of woman. Okay, very simple children. This might be asked for 1 mark. Next, mention any 4 tools used for tracing the evolutionary relationship to study the human evolution. Very important. Tools. So when I ask tools, immediately write this answer without thinking. The tools which are used to trace the evolutionary relationship between the human beings. That is very important question. The answer is, first tool is excavation, second tool is carbon dating, third tool is study of fossils, fourth tool is determining of DNA sequences. So they say determination of DNA sequences. So these are the 4 tools which are helping in tracing the evolutionary relationship. So next, 14th question, how do genes control the characteristics of tallness of a plant? You must understand. See what is meant by genes, where are the genes, all these things I have already explained in the crash course. Okay. See, these genes are present in the chromosomes. Okay, they determine the trait of that particular organism. Here, the genes are controlling the characters of a tallness of a plant. Why? See, it is because the genes for the tallness will give instruction to the plant cells to generate many plants' growth hormones, due to which the plants will only grow tall. Okay. On the other hand, if the plant has the gene for shortness, so less plant growth hormones will be produced, because we are focusing on the tallness here. Okay. So due to which what happened, the plant will not grow much and it remain a dwarf plant only. Hope you understand. So that how do genes control the characters is a very important topic. You must know it. So moving to next question. Only variations that confer an advantage to an individual organism will survive in a population. Very important children. See, only variations that confer. But what is the, in simple words I say. See, which organisms is accepting the variation, that means showing the changes in the environment, can easily survive in the environment. It's a very simple way of understanding the questions. For example, if my body is not capable of, adjusting to the environment where there is more snowfall, then I will die very fastly, because my body is not accepting that particular condition. So only if, organism shows variation, that will give an advantage for that particular organism to survive in the environment. Okay. So the answer is, only organisms with the variations are indeed considered as an advantage, which proves useful for its survival in the given population. So the extent to which an organism will survive depends upon the nature of the variation. Take an example. If you take bacteria, so may withstand heat waves due to its genetic variation, but not in the cold environment, then the same bacteria may not survive. It is surviving in the hot condition, but it is not able to survive in the cold condition. Then all those organisms get vanished. Okay, so it means the genetic variation in the bacteria is an advantage. So only in the case of hot environment it is surviving, but it comes to the cold environment, it will die. So this happens due to the lack of better adaptive skills for the cold environment. Very important. So you can alter this answer and write in your own way also. It is not that mandatory that how I am explaining here in the same way you have to write. Okay, understand the concept. Write your answer in your own method. Understand? But let the answer be in the point wise. Don't write like a paragraph. So moving to next question. The tall pea plant bearing the red color flowers, okay, is crossed with the dwarf pea plants bearing the white color flowers. This particular one also I have explained in that video. You can check out. See children, it is same as how do you do for round yellow and wrinkled green. So the parents here are tall, right, that is capital T, capital R, and the dwarf white. Okay, so small t, small r, smaller. First what will you do? You will cross pollinate. When you cross pollinate in F1 generation, you will get capital T, capital R, small r, which is a tall red as they have dominant traits. So what will you do next? Next you will self-pollinate the same traits on segregation. After segregation you will self-pollinate. It is similar to round yellow and wrinkled green, but only the traits are being changed here. Tall is representing the height, red is representing the color. There are two different traits. If there are two different traits with their contrasting characters, we will call it as Mendel's dihybrid cross. Okay, or sometimes it also can be called as hybrid cross. Okay, you have to note down. So here also on self-pollination, we get the phenotypic ratio same, that is 9 is to 3 is to 3 is to 1. So 9 tall red, 3 tall white, 3 dwarf red and 1 dwarf white. Okay, it is same as the dihybrid checkerboard you write for round yellow and wrinkled green, but the alleles are different. Okay, same method you follow. Moving to next question, differentiate between acquired traits and inherited traits. I told you. So the traits which are developed in an individual due to the special condition, I say it as acquired. For example, see, if I want my hair color to be changed, I can go to the salon and I get my hair color changed. It is not that my progeny will get the changed hair color which I have now. Okay, because the alterations has been done by me during my lifetime. Okay, so this is what we will call it as acquired traits. But when we talk about inherited traits, these are the traits which are passed from one generation to another generation, from parents to you to your progeny. Okay, so they cannot direct the evolution. That is, acquired traits, they cannot direct evolution, but inherited trait can show the evolutionary changes from one generation to another generation. So example for acquired traits, you can take the building the body muscles, or you can take hair color. So all these things are example for acquired traits. Inherited trait, we can take free earlobe, okay, attached earlobe, color of eyes, and shape of the foots. All these things are example for inherited. So next question, briefly explain about the artificial selection with respect to evolution of wild cabbage. Most of you will forget to study about artificial selection because it is at the end of the lesson. See, dear students, when we talk about evolution, you must know all the evolutions. Okay. So here, when I talk about artificial selection, okay, humans have been a powerful agent in modifying the wild species to suit their own requirement through the ages by using the artificial selection. Why do we call it as artificial? Because it is happened with the involvement of human beings. Okay. So what do the earlier farmers did was, there was a wild cabbage. So those farmers want the wild cabbage in a different appearance. Okay, so they took the wild cabbage and they start to cross-pollinate the pollen grains of those wild cabbage plant with a variety of plants as per their needs. As a result of it, from the wild cabbage, they got many varieties like broccoli, cauliflower, red cabbage, kale, kohlrabi. So these were the different variety of, the vegetables which they have obtained only from the wild cabbage. Wild cabbage is the ancestor for all these things. Understand? So when they ask about artificial selection, you give this example. Okay, it is given in your textbook. Do not forget. And all these are broccoli, cauliflower, cabbage, kale, kohlrabi. They are all the different forms of the wild cabbage. Ancestor is wild cabbage itself. And this is done artificially because with the human involvement, all these vegetables have been came to existence. I hope you found this video useful, dear students. I have explained 18 important different concepts from the lesson heredity and evolution, and all these concepts are very important. Repeatedly they have asked in the examination. You can expect either checkerboard or you can expect the questions from the acquired and inherited traits, or you can expect from the principles of Mendel's principles. See, Mendel's principle is also very important. So when I talk about Mendel's principles, so he has come up with the principle of dominance, principle of segregation, and principle of independent assortment. Okay, so I have already explained these concepts in the crash course. Okay, so whatever the concept which I have been conveyed to you in this particular video, please, you have to concentrate and learn the answers for this particular questions. Okay, so see you all in the next video series 7. Until then, take care. All the best for your examination. Do not waste your time and take out all the videos which I have posted in the YouTube and utilize in a maximum way.